Okay. Thanks, everyone. Hey, welcome to the Research Innovation Spotlight series on tools and diagnostics. We're joined today by 4 amazing researchers going to present today, and we have also our esteemed moderator, Kathryn Zavala. This program would not be possible without the support of Alexandria Venture Investments, and thank you, Hallie, for all your support. And we also do this in collaboration with Bioscience LA. And Dave, do you want to say hi briefly and talk
I'll say hi very briefly, but yeah, great to see everyone. And Dave Whalen, CEO of Bioscience LA, growing the life science ecosystem here in the LA and SoCal region, and working very closely with the Alliance, and actually working really closely with MedTech Innovator and Biotools Innovator. So thrilled to have Kathryn here as our moderator.
Great, thanks. So the next step, I will actually have the attendees introduce themselves, and then I will leave it to Kathryn, and she'll kick things off. I'll just call on you, then let you know who's next. I'm sure some people will start filtering in a bit, but for right now, well, Hallie, let's start with you.
Hi, nice to see everyone. I'm Hallie Kuhn, VP of Science and Technology from Alexandria Venture Investments, and oversee our corporate venture investments in Southern California.
Okay, thanks. Benji, would you say hi?
Hey everyone, my name is Benji. I'm originally from the La Jolla area. I now unfortunately live in Chicago. I work for an early stage venture fund called KDT. We invest at the intersection of like biology, computer science, and compute. Very nice to meet you all.
Thanks for showing up. Hey, Lauren, you're next.
Hey, everyone. I'm Lauren Bradley. I'm the Associate Director of Corporate Engagement at the Jackson Laboratory. We are a nonprofit biomedical research institute that specializes in genomics and genetics, as well as specialized mouse models. Our institute is based in Bar Harbor, Maine, but I work remotely from Los Angeles. Happy to be here.
Great. Thanks, Lauren. David Pearson.
Good morning, everybody. David Pearson from UC Riverside. I'm the Managing Director of Entrepreneurial Programs in the Office of Technology Partnerships. My fun job is that I run the Life Sciences Incubator at the university. We now have 10 companies in doing research.
All right, thanks, David. Ryan, if you are available, please introduce yourself. It's working on it.
Sorry, I'm moving into a different room. So I don't know if you can see me, but I'm Ryan Witt. I'm the Hub Director at UCLA's Biodesign program. I'm also a member of a group called Tech Coast Angels, which is a local angel investor group.
Great. Thanks, Ryan. No, we cannot see you, but I know what you look like, so no worries. All right. Great. Well, with that, I will pass this off to Kathryn Zavala, who is the Managing Director of Biotools, as well as COO of MedTech Innovator, right? So I'll have it. You do the rest of your introduction. I think you've got a little bit of a show and tell.
Yeah, no, great. Thank you, Steve. Really appreciate, you and Dave and all the work you've done to bring us all together. I'm very honored to be the guest host today and introducing some of these amazing innovators that we have in our SoCal area. I did want to give a little bit of context of, why I was invited to host this. So as Steve mentioned, I'm the COO at MedTech Innovator, but more recently, I'm now the Managing Director of our new program called Biotools Innovator. So Biotools Innovator, we launched last year as the first nonprofit accelerator in global competition, solely focused on life science tools and diagnostics. And so, what does it mean to be powered by MedTech Innovator? Our organization has been around since 2013. This is our 10th year in operation. We've graduated 421 alumni that have all gone on to raise $4.3 billion in follow-on equity funding. So as a nonprofit, none of that funding comes from us. We do award grant funding, but this is all outside investment. So really good testament to the quality of companies that we've seen. And additionally, that has led to 24 acquisitions. So we've taken that great model that we've seen work for medtech, and we're transitioning that and focusing that on the life science tools industry. And so really we've built this accelerator with the sole purpose of accelerating innovation. And so, we provide resources, as I mentioned, the non-dilutive funding, visibility. So we have some of the biggest stages in the industry where we can highlight the innovators, a sense of community. So as being a nonprofit and startup ourselves, we understand how important it is for innovators to interact with each other and learn from each other. And lastly, mentorship. And so the mentorship component is hugely important. And to drive that, we build these corporate innovation networks or ecosystems. And so, as I mentioned, MedTech Innovator is our biggest, longest standing program. We also have a program in Asia Pacific, a pediatric focused program, and then, as I mentioned, our newest one, which is Biotools Innovator. So all the logos that you see here obviously support us financially from a nonprofit perspective, but equally and probably more importantly is they work one-on-one with the startups in our portfolio to make sure that they're able to succeed. And so just wanted to give that overview of where I come from and the kind of the ecosystem that we're operating in. Today's focus is a little more on that earlier stage part. So many of our innovators here work in an academic setting, but they're developing technology that has both the potential to be licensed out, spun out, or part of industry-sponsored research. And so between Dave, Steve, and I, we landed on an overall theme of neuro. So you'll see a very I guess a varied amount of technology. So I think I don't think anyone's overlapping. So it should be a really fun day.
Hey, Kathryn, before you go forward, I just realized I skipped Andy, and then we also have a new attendee who just popped in. I'll make sure to get a chance to introduce themselves. So sorry,
course.
Andy, I apologize. We introduced Gandalf earlier, but forgot you. So please take the floor.
Well, we didn't miss the most important participant. So I'm Andy. I'm the executive director of the Alliance. Excited about all the amazing things happening in SoCal and appreciate what's happening on the device and medtech and biotech side. So it's always exciting to be with all of you. And Gandalf is excited to participate as well.
Yes, always good. And Kathleen.
Hi there, I'm a Senior Director in External Innovation in the Delivery Devices and Connected Solutions organization within Eli Lilly.
Great, thanks for
coming.
Great, all right, Kathryn, it's back to you.
Yeah, well, I mean, I think this is a very excellent group. And, as I think some of you have participated in this before, but for anyone new, the intent is for it to be very interactive, for you to ask questions and learn a lot more about the researchers that we're highlighting today. So with that, I won't take any much more of the time. I want to start with our first presenter. So that's going to be Dr. Akbari from UCLA.
Thank you, Kathryn. I'm going to start sharing my screen here. Can you guys see my screen? I'm going to go into presenter mode.
And I apologize. I said UCLA. I meant UC Irvine.
Yeah, and I'll just start with a little quick video that'll also introduce myself.
With the arrival of portable, affordable, and multimodal neurodiagnostic devices, the complex structure of the brain will be brilliantly revealed, and this will help greatly in the treatment of many brain and body-related trauma and disorders. BMJ 2020, the future of brain and body health. What would the face of medicine look like if parents, caregivers, first responders, and the general public could diagnose and facilitate the treatment of brain and organ trauma? Well, at Waveflow, our efforts to make this a reality have already been tested through the participation of leading physicians, technicians, and professors for the past 8 years, resulting in 7 pending and approved patents. Under the guidance and supervision of Dr. Yama Akbari, a physician and scientist at the University of California's Irvine's Department of Neurology and the Beckman Laser Institute, researchers have applied neurodiagnostic technology in acute brain injury and have identified significant solutions that have resulted in several published papers and government grants. By working with Waveflow, the Akbari lab brings together an expert team of clinicians, scientists, and engineers focused on taking technology from the laboratory to the clinical setting to help impact the lives of patients. One of Waveflow's devices, which aims to make actionable diagnostic data available to everyone in near real time, is our new Waveflow Pin. The Waveflow Pin is being developed as a pocket-sized, multimodal medical monitoring device that combines electronic, photonic, and magnetic data. Our device can read a person's brain waves, blood flow, and oxygen metabolism, all in near real time. Traditional EEG devices can directly record the electrical activity of the brain. They have proven to be indispensable as a tool for the diagnosis and treatment of epileptic seizures, brain trauma, concussions, and other neurological conditions. The Waveflow Pin takes EEG technology to a new level by uniquely combining a small handheld EEG device with photonic and magnetic technology to enable additional trauma diagnosis such as strokes, brain hemorrhages, cardiac arrest, COVID-19 therapy, and drug overdoses, while also enabling rapid treatment using stimulation modalities. With our PIN's aggregated matrix of actionable data, our goal is to help patients and doctors make more informed, near-instant, life-saving decisions. Brain diagnostic exams usually take place at a clinic or hospital using large commercial EEG machines. Now with the Waveflow Pin, we can eliminate the excess time, effort, and cost to acquire such readings. Readings can be quickly and easily taken outside of the hospital setting and viewed on your smartphone or tablet. When it comes to the diagnosis and therapy of organs damaged by diseases or viruses, the Waveflow Pin can empower pharmacies with more constant, flexible, and affordable monitoring to adjust medication and therapy. We at Waveflow are also leveraging an exclusive development relationship with StoreShare, an enterprise-grade compliant cloud system. StoreShare is a patented compliant HIPAA storage platform designed for the archiving and sharing of medical data. StoreShare allows families to manage their health and well-being. This is achieved through StoreShare's data conversion platform machine, human-readable files and reports, and a platform for hundreds of medical and performance apps. Affordable and easy to use, the Waveflow Pin gives access to accurate, near real-time medical data that could save millions of lives. Take control of your family's health with the Waveflow Pin.
So Waveflow is a company built out from UCI that develops optoelectronic devices for diagnostics, therapeutics, and performance of the brain and other organs. As a physician and scientist, I take care of patients. I'm a neurologist by training, specializing in neurocritical care, and I have a research lab, an animal research lab on the main campus. And so I work with several biomedical engineering professors and physicists on the main campus. And then I go back and forth between the hospital and the laboratory setting. And so these have ended up with several IP from our group. And these have been more recently than sponsored by Mark Nurzai. And along the way, we've had several people join our team. Tyler Urani, as well as 6 of our students actually who've been with us for the past year now, who are actually finishing up their biomedical engineering degrees next month, who've been very important in the prototype design that we have for the initial device that I'm going to be talking about, and that was mentioned a little bit in the video. The problems that we're focused on is bringing the hospital's services into the home setting, taking complex hospital machines into a portable device, taking very expensive machines, making them much more affordable, that can offer real-time diagnoses, and which can enable rapid therapeutics as well. And so it's essentially an all-in-one device And we're trying to really overcome a lot of global healthcare disparity that I feel that is worsening. Our device is not intended to replace a lot of these complex machines, but certainly it can get the job done faster, quicker. It can make a significant difference in where, especially in the setting that I work, which is the intensive care unit, where I see a lot of suffering. Over the years, I've really taken it to heart that we need a device like this that can really make a huge impact, because time is brain. And so our main device is essentially a pen EEG, and it includes a matrix of EEG blood flow, metabolism, as well as oxygenation that can then be displayed on a smart device. The current prototype that we have can read a wide spectrum of brainwave frequencies. And so by that, I mean, there are different types of brain waves that we have, that some are slow, some are fast. They each are important for different facets of our cognition and our neurological activity. And we use EEGs on all of our patients, essentially in the intensive care unit, in the neuro-intensive care unit. And so that is lacking in many domains within and outside of the hospital setting. That can be rapid. And so the prototype that we have has a solid contact with the head, retractable legs, very comfortable, portable, and pocket size. And those are some of the key things that are differentiating our device from the bulky ones. And of course, the other modalities that I mentioned, as far as the optical features, they don't exist in combination right now. And so the acquisition module that I just showed you incorporates dry electrodes, although we can use wet electrodes, amplifies the circuit, filters it, converts it to a digital signal, and then using a microcontroller transmits it to a mobile device, which converts raw data into a filtered data set that can then be accessible by anybody anywhere through a cloud system. The design is pretty simple. Using 3D modeling software, it can be modified. And that's important because we want to customize it to the needs of different types of consumers, whether it's physicians or non-health personnel. And that goes to the global market. So aside from healthcare personnel, which includes diagnostics and therapeutics, there's a large market for this in sports performance as well. So not only do we want the parent of a patient, of a child with seizures or a medical condition to be able to use this at home to help their family members. Not only do we want it for physicians or paramedics or nurses, but we believe this can be heavily used actually in the sports performance field as well. The focus by year, the market focus is essentially over the next 1 to 2 years, a year and a half or so. We're looking for 510(k) approval process for the diagnostics side of it, starting with the electronics, which we have the working prototype of, and building the optical features as well, remaining diagnostic mostly for the first couple of years thereafter, after which then we will then incorporate the next phase, which would be a therapeutic phase, which will then transition into performance usage as well. Financial projections are modest here, but it certainly will take time to build up to what we envision can be a pretty large market over the next several years. We're in the process of building our team, recruiting additional people to help us accomplish this. And so the competition is there, but we have a lot of solutions that can help us overcome the competition. So taking large EEG devices, making them portable. There's certainly a lot of EEG companies out there that use headsets or small devices, but ours has specific features that will help the consumer. And I'm going to be first in line to use this device on my patients to try to help them in a rapid way. And so these are things that I've used a lot of these devices that are listed here on the left side, and I've seen numerous limitations in them, and that's why we're trying to make this a solvable problem. The oximetry-based devices, they're not multimodal. They don't combine perfusion metabolism. Ours does. We're using a matrix of combining EEG, blood flow, metabolism. This is something that we have had established in our research lab on animals over the past several years. It does not exist in humans, and that's what we're trying to make that transition from animals to humans. Photobiomodulation is out there quite a bit. We're trying to make that more compact. And then electrical magnetic stimulation is out there, FDA approved products, but we're combining it with this matrix of information that we have to make it a combined a portable unit that will allow closed loop transitions. Our sales channels are going to be targeting pharmaceuticals, hospitals, B2B, as well as military, retailers, B2C, and of course, sports medicine. The business model is focuses on not only the affordable device that we have here that I've described, but also a monthly monthly subscription for the software suite, so that there can be cloud-based resources such as physicians, neurologists, and other people to help with interpretation of any signals that may be necessary, and a constant service for the people that are using it. And so the pipeline is as such. We're here right now with the 3D product design and testing, mostly on the electronics. We're transitioning into the photonics as well, which will include additional IP and the 510K and then final device. And a lot of this is going to be in phases as we speak. And so that's pretty much it. I'll stop there, and happy to answer any questions.
Great. Well, thank you for that presentation, Dr. Akbari. So we'll open up the floor for questions. So either people can just jump right in, or they can use the hand gesture to ask a question. So I see Ben has a question first. So we'll go with you, Ben.
Awesome. So great presentation. Really enjoyed that. Thank you. I guess my question is around, so your initial 510K approval, what indications for use are you looking for?
So rapid diagnosis of seizures. There's one of the most frequent consults that we get as neurointensive care physicians is altered mental status. And so when we have an altered mental status person, or somebody in a coma, for example, which is essentially what happens when a paramedic approaches a person that's passed out, on the beach or something like that, right? They could have suffered a stroke, they could have suffered a seizure, they could have suffered an opioid overdose. We don't know what it is, right? And that takes time for them. And usually what happens in those situations is that you check for a pulse, you give some oxygen, you take them to the hospital to figure out what the heck's going on. And so the simplest diagnosis is seizure, no seizure, stroke or no stroke. Stroke gets a little bit more complicated because there's ischemic and hemorrhagic and whatnot. But essentially, activity of the brain or no activity of the brain. Those are all simple indications that I think would be widely accepted.
Great question. I guess I have a question about the underlying technology. So I know that you mentioned that you have 7 patents already for that. So what is, I guess, the uniqueness about the technology? Is it miniaturizing something? Is it combining a bunch of different technologies?
Yeah. So the IP is focused. One of the IP is focused specifically on the device itself. So that is correct. The key thing about the IP is the combination of the flow and metabolism that it has. And so through our animal experiments, we've shown that by combining the blood flow in the brain and the metabolism in the brain, as well as the electrical activity, you can have unique signatures that can help diagnoses. So that's where the IP is. When you focus on the IP, I'll give you an example. For example, when a patient has cardiac arrest, there are, I sign probably more death certificates, unfortunately, than the vast majority of physicians. I used to think death is a very static process. And what I've learned through my research is that it's not. When we're dying, it's actually, the brain is immensely dynamic. There are these strange waves, these brain tsunamis that occur in the brain as we're dying. And a lot of times over the past few decades, these were called waves of death, but they're not waves of death. Actually, they're waves of opportunity, because our research lab is one of the few labs that can actually resuscitate animals to come back. It's very easy to capture death or euthanize an animal and we do some research, but our focus is to bring the animals back. And when we do that, we see all these unique waves happening that we normally cannot see. And so our device actually captures those waves that we normally miss. And by combining the EEG and the blood flow, we can actually predict. So for example, during death, we have one way where we can predict up to 90% the outcome of an animal, how good they are. And it helps us guide therapy. How much blood flow should we give to the brain during CPR, after CPR, in the moments after CPR, during a stroke that's happening and after a stroke. And so the IP goes into the details of combining blood flow metabolism into something called neurovascular coupling or flow metabolism coupling. And by using specific metrics, specific equations that we've come up with, we're able to tailor the flow, which is essentially the supply, the blood supply, to the demand, which is the metabolism. And if you have too much supply, you get injury, brain injury, it's called reperfusion injury. And if you give too little blood supply, you get ischemia. And so we're trying to match that blood flow to the supply to the demand. And that's exactly what the device allows us to do while monitoring the electrical activity as well.
Interesting. So that last part of it kind of speaks to when you talked about like the therapeutic potential. So not just titrating, for example, a drug therapy and saying where people are, but actually delivering a therapy to the brain as well.
That's right. That's right. So some of these treatments that we have that can actually be given pharmacologically can actually also be given electrically through stimulation or through optical features such as a shock wave, or magnetically, giving a pulse of magnetic stimulation. And so research over the past 10 or 15, maybe 20 years or so has shown all of these technologies are FDA approved, actually. Magnetic stimulation, photobiomodulation, they're used for different purposes, for pain, for other indications. Our focus is more acute, severe, injury, and these using a device, so a non-pharmacologic way, can actually induce many of these traditionally pharmacologic, in a much faster way.
And also, I mean, do you see it potentially augmenting pharmacological therapy?
Definitely, and that's the key, because every patient, I mean, ultimately we have to go to what we know the best, and over the past 100 years of medicine, we know that pharmacological therapy is really the standard of care. And so a lot of times we don't know. And a perfect example of that is actually when we have to do CPR on patients in my intensive care unit, whether if you're 100 pounds versus 300 pounds, the guidelines recommend 1 milligram of epinephrine. That seems kind of silly. 100 pound person versus a 300 pound person, they shouldn't get the same amount of a medication. It should be tailored to and right now it's not. And so with this kind of device that can be used even during CPR or other types of emergencies like a stroke happening in real time, we can give the proper amount of medication to tailor to the brain. And during cardiac arrest, I should mention that the brain is the number one determinant of outcome, because we can bring people back from cardiac arrest, but usually their brain is not well.
Well, that's great, Dr. Akbari. I think this is a really good way to start off today's forum. Seems like there's a lot of potential there, as you mentioned, diagnostic, therapeutic, and performance. So I want to move on to our next presenter, but before that, I want to a round of applause for Dr. Akbari. Great presentation.
Thank you very much. Thank you for having me here.
Okay, so we'll move on to our next presenter. So Dr. Freedman from UC Riverside.
Hi, everyone. Let me share my screen. Do you guys see it okay?
We do.
Okay.
Yeah, you want to get a So we see the notes right now.
Yeah.
Okay.
Okay, gotcha.
There you go.
Awesome. Thank you so much for the invite to present. I don't have a slide on my background, but I actually got my introduction into research was looking at epilepsy. So I do come from a somewhat neuro background. So we're looking at, intracranial recordings, doing single cell measurements of neurons, and looking at predicting when a seizure might happen in the future, and then stimulating the brain to stop it from ever happening. So that was kind of like my introduction into research. And then I was just fascinated with the brain and how signals are generated. So I got interested in nanopores as the foundation for neuroscience. And so, yeah, I fell in love with nanopores. And for better or worse, I've been doing that ever since. So I'm going to be talking about nanopore sensing as it relates to diagnostics and drug delivery. I can't talk about nanopores without introducing, the driving force behind it, which is DNA sequencing. And so, we've seen the cost of a genome come down quite significantly, especially when there's some injection of a new technology. And so we're hoping that the next big injection of a new technology will be nanopores. And it's really this tenfold or so decrease that will really make this technology go into like a local doctor's office, for example. And so just to show that we can get base information, this was some early work, not from my group, but showing that individual bases can give like specific information or be read out by the nanopore. So that work was done using alpha hemolysin. I don't work with this pore anymore. I use solid state nanopores. So these are structures that are made out of silicon, most likely silicon-based materials, either silicon nitride, graphene, 2D materials, stuff like that. And so we engineer these to be any diameter. In this particular case, these have the same diameter of around 2 nanometers. And so what else can we do? Obviously commercialization. This is Oxford Nanopore's MinION, which is a very small USB-based device for DNA sequencing. And this is now, on the ISS. I do, much broader usage of this nanopore device. I've looked at biophysics, looking at proteins. This was some early work showing that the electric field inside the nanopore actually unfolds the protein. And so we can look at, pathology processes by looking at these single molecule events. Okay, and so most diseases start off oftentimes with a single molecule. And so the sensing mechanism is based on this indirect volumetric occlusion principle. And so for an open pore, this is KCl, but you can also use, NaCl, which is just table salt. And so when you apply a voltage, you get this transport of these ions, and this generates ionic current. And so when a molecule is inside the pore, you get a blocked flow of ions. In reality, there's a lot of nanoscale physics involved, which I'm very interested in. And so most cases, the driving force is that the DNA gets pulled through electrophoresis, but there's really other forces as well, electroosmotic force, which is based on the fact that you have a charged surface. This is true for both silicon nitride nanopores, which is probably the most common, or glass. And then my lab recently has been looking at how we can apply pressure as well to, get more information from these molecules. And so why do we use nanopores when there's all these other single molecule techniques? Well, first of all, it's definitely the most high throughput single molecule sensor. And that's because the sensor isn't moving. It's the molecules that are driven through the sensor. It's label free. So, you're measuring them in a fluid. You don't have to modify it in any way. You can produce them very cheap. And best of all, there's no optics. It's all electrical readout. So what are some applications? I'm going to be talking today about femtomolar sensing. This is really applicable to liquid biopsy. So this is the idea that, maybe you can't get at the tissue that has some malignancy or pathology, but it's spewing out molecules into the bloodstream, which we can then biopsy and take and measure. And so this is really beneficial for early phase diagnostics or at-home testing. And so one of the areas where this is being used is to detect ALS, looking at this amyloid beta peptide fragment assay, which has a very minor change, 2 amino acid difference. And so this is typically done, this assay is done using mass spec, which has its downfalls. So can we use nanopores to detect this? Of course, DNA and protein sequencing, discovering patient-specific variations, lipid particle characterization, DNA data storage is kind of a topic that's emerging, and then biotech for space travel. So I'm going to be talking about mainly these two things. So in terms of liquid biopsy efforts, my lab has been doing a lot with pre-concentration of a target molecule. And so the idea is to accumulate low abundance molecules, taking note that the bloodstream is fairly large, 6.5 liters, and it's very well mixed. And so we're not talking about detecting molar quantities, which is 10 to the 23rd molecules, but can we detect hundreds of molecules and make some sort of assessment about disease state? So getting into sort of the hardware that we use, silicon nitride is a common type of architecture that we use. We do some work with this. For rapid prototyping and proof of concept work, we typically use these glass nanopipettes. If you've ever done electrophysiology, you probably are familiar with these. But we can make nanopores that are down to 5 nanometers with this benchtop device. And so this was some somewhat older work where I coated the nanopipette with gold and applied a secondary force onto DNA. This is called dielectrophoresis, and we're able to capture DNA at the tip of the sensor and then translocate the DNA for sensing applications. And so again, we kind of apply this AC voltage to do the capturing. I won't talk too much about the mechanisms involved. And then after we accumulate the DNA, we pass it through the pore. And so this is the current best in terms of concentration. We can go to attomolar concentrations. Here, the lowest is femtomolar, but we can do attomolar as well. And we can get, control the capture of molecules. We've also, since then, this is more recent, shown other trapping mechanisms. In this case, we have trapping occurring inside the pipette, and we don't have to functionalize this with gold. There is no, specific design that we have to do. And so we can capture DNA. In this case, this is fluorescently labeled PCR fragments. And so we get this trap that occurs. And so the idea is that we trap and concentrate DNA, and then here we reverse the voltage. So positive voltage kind of traps and accumulates, and then negative voltage releases this DNA. And so we get this, initially the DNA is sensed very slow, like less than 1 molecule per second. And by doing this trapping and accumulating, we can get, a huge release of DNA. And so there's this enhancement that we get to achieve. Okay, and so that's what I wanted to talk about that. We also are doing characterization of lipid-based particles, and these could be endogenous, so exosomes or extracellular vesicles being spewed out from, a tumor, for example, typically are well vasculated and spew out these lipid vesicles, or these could be drug delivery vesicles or liposomes. And so some of the more characteristic features are size, volume, charge. Some of the more new properties that we've been able to measure are lipid flexibility or deformability. What are the internal contents of these liposomes, and self-adhesion properties. Okay, and the way that we do look at these types of particles is by using a pressure wave. So we have a pressure wave that oscillates between vacuum and pressure, and we get the ability to recapture the same particle over and over again. So we capture it here, it switches, and then we kind of spit out the particle again. And so we did calibration with microspheres. We can actually measure populations, microbial populations. And we eventually found out that we're actually measuring mass, or really mass charge ratio, due to the fact that these particles have to enter the pore, decelerate, and then accelerate again. And the lag that is, the time lag that is introduced is a function of both mass and charge. And so we get this ability to recognize different populations. And this is some COMSOL, some numerical modeling that we've done to validate this. And so as far as mass sensing, it really comes about due to the very high G-forces that are experienced by the particle inside the pore. One of the most sensitive mass sensors is QCM, which is about a 1 million G. And so we're not quite that far, but we can measure some interesting properties of these things. So we've been looking at both synthetic lipids for drug carriers as well as RBC derived, so red blood cell derived, which produce a lower immune response. Interestingly, there's not too much in terms of measuring concentration of these RBCs because their composition is unknown. So we can use nanopipettes to measure concentration as well as some other properties. So we've been looking at these, characterizing these particles. And so we can look at hydrodynamic deformation. We can look at the transport properties, basically how long this particle spends inside the pore. So we call this the dwell time. And so we can look at empty versus drug-filled nanoliposomes and see a difference between them. Also, how long it takes for the particle to enter the pore and then come back out, which is essentially the recapture time. We can also see a difference between drug-filled and empty nanoliposomes in those two samples as well. And then this is my last slide. We're also showing that we can measure deformation and actually self-adhesion properties of cells as well as nanoliposomes. And this is based on the fact that we can measure the same entity more than one time. And so, this is pass 1 and 2. We start out with a single aggregate. And as we apply these forces, it splits up into 3 different particles. Okay, and so we can look at deformation, lipid properties, what are the mechanics of the lipid membrane as well. So this is a lot of the students and postdocs that have done this work. And yeah, I'll be happy to take any questions you have.
That was great. Thank you, Dr. Freedman. Obviously, powerful technology with a lot of potential uses that you have there. I'll kick off the questions. The first one, you mentioned specificity, antibody binding and aptamers. Are there any other ways you can identify molecules using this technology?
Yes, so obviously the nanopore is doesn't tell us about the identity. It's based on this indirect volumetric occlusion principle. And so, yeah, we can use antibodies to bind to different targets and measure the increase in size. Let me see here. So yeah, we can, we both do direct aptamer attachment, or we can also do aptamers bound to what we call molecular carriers. And so these are DNA. DNA is very suitable to pass through the pore. It's just very well behaved. It's negative. It's homogeneous in its charge. And so we can attach aptamers that can bind to different target molecules, and we can measure the corresponding electrical signals. We can also look at Cas9 binding. So Cas9 is quite nice because you don't have to open up the DNA strand, the duplex of the DNA. We can do like DNA domain detection using Cas9 and program it using the guide RNA. So we can look at, does this DNA domain exist in the sample? Also, in terms of Cas9 therapies, looking at off-target effects, where does Cas9 bind? Because ultimately, where that Cas9 molecule binds is where it's going to cleave the DNA strand. And so you can do some characterization of the Cas9 therapy. And then, yeah, you can use antibodies to also bind different targets and measure the corresponding electrical signal. And we have done these studies in, a very, mixed sample. So fetal bovine serum is has a lot of other proteins in there. So it's a very heterogeneous sample. And so we're still able to do the detection even in serum samples.
Great. I see Ben has a question.
Yeah, thank you. So great presentation. I think that, one thing that my firm has been looking at in the protein sequencing space is how nanopores can handle post-translational modifications. So if you kind of curate a set of short peptides, like, yeah, you can probably sequence it because it's linear. But the question is, like, what happens when you start introducing like these disulfide bonds and these sialic groups and just these bulky additions to the protein structure? Like, how does that affect, your ability to pull the protein through at a constant velocity?
Yeah, so DNA or protein sequencing is definitely a huge challenge, and yeah, we're just starting. Like the nanopore field is kind of just entering that field in the past few years. Disulfide bonds, I think the common approach and what we do in my lab is to break the disulfide bond, and so we do that reduction to make it more linear. And so yeah, I don't foresee. If you have a loop or the protein is folded because of that disulfide bond, I mean, I don't see how you can do sequencing of it. You really have to make it linear and then pull it through the pore. As far as the other, you mentioned some bulky groups on there. Yeah, that's a tough challenge that I don't think anyone has a good answer for. As far as making the velocity of the peptide constant, that's also a big challenge. What we do in my lab is to kind of use SDS to make it homogeneously negative. And we're not sure if that is going to end up working or not, but yeah, it's a good question, but I don't think anyone has the answer.
Thanks, Ben. So another question I have for you, Kevin. So, there's obviously limitations to what kind of molecules can be sensed. Are there any other kind of restrictions?
So really, it all comes down to signal to noise. So if you have a pore that is, say, 250 nanometers, it depends, you don't want a molecule to sit and clog the pore because that'll, you can typically unclog it using various techniques. However, assuming that all the particles are less than this diameter, you start to lose signal to noise when it's about a quarter of this size. So a quarter of 250 nanometers, the nanopore won't really give you enough signal to noise to measure particles that are, 50 nanometers, for example. You just won't get the signal to noise, but you can always modify the pore size. That's the benefit of solid state nanopores, is that you can modify this pore size to whatever you want it to be. So yeah.
And in the spirit of, kind of today's forum, what are some potential kind of like partnerships or industry sponsored research? Like, how do you want to interact with others and with this technology?
Yeah, I'm interested in, industry sponsored research mostly. I think a lot of this work is obviously very preliminary. We do proof of concept work in my lab, so we develop a technology and usually patent it. This has a patent, this concentration way of concentrating DNA at the tip. Also, this mass charge sensing has a patent pending. So, but there's a lot of other things that my lab does. We kind of sense anything that can fit through the pore. And so there's lots of applications. And so, yeah, I would be happy to work with the company to help build, new diagnostics and yeah, see where it goes.
Excellent. Well, thank you, Dr. Freedman. So another round of applause. Great presentation.
Thank you.
Very intriguing. Okay, so let's move on to our third presenter. So this is Dr. Ozcan from UCLA.
Hi, everyone. Are you able to see my slides?
They're just coming up. Okay, so we can see all your slides. You'll just want to, there you go.
Awesome. Well, thanks for giving me the opportunity. So I'm a faculty at UCLA School of Engineering, and my work is on the use of light for computational microscopy and sensing. So today I'll talk to you about some of our work on virtual staining of tissue using deep learning tools. My lab was one of the first groups to utilize the power of deep neural networks for image reconstruction, for image transformations, especially in microscopy space. Of course, machine learning tools are great for example, image annotation, classification, tumor margin detection, etc. But I think they also hold tremendous potential for using them as part of actually an imaging system to do image reconstruction and change the way that microscopy or sensing operates at the hardware and software fusion. And as I said, we were one of the first groups to apply this to microscopy. In fact, the first group, and we have an issued patent on deep learning microscopy, portfolio of them actually licensed already by some of the leading microscopy companies. And what makes deep learning so special is that it enables cross-modality transformations. What I mean by that is one form of microscope, let's say an inexpensive microscope, can actually be working like a more advanced diffraction-limited microscope through establishing a statistical transformation from one form of microscope to another. And there are tremendous applications that can utilize these kinds of image reconstruction, image enhancement modules. And I'll just share with you one that touches on virtual staining. So this is going to be a cross-modality transformation where the input images to the neural network will be fluorescence microscopy, in fact, autofluorescence microscopy images. And through a neural network, we want them to look like bright field microscopy images of the same sample. So this is a very difficult to establish transformation because we do not know the link between fluorescence or autofluorescence and bright field. These are two different quantities. And there's not a sufficient physical insight of light matter interaction that's going to enable us to do this transformation. But that's where data, microscopically registered data, would be crucial. All right, so that's one of the transformations that we want to achieve, fluorescence to bright field. As if this is not challenging enough, we would like to also make this transformation even harder by having the inputs to be label-free, and we want the outputs to be labeled. So the microscopy modality is going to be changing, and the sample itself is going to also go through a transformation of being label-free and then labeled. So of course, if I can achieve this kind of a transformation, I can change the history of histopathology in the sense that we can now, perhaps with this kind of a transformation, attempt at doing a different way of pathological workflow in our clinical histology labs. So, for the last 150 years, literally for the last 150 years, every time we take tissue out of the body, it goes through this laborious histochemical staining process with chemicals, oftentimes chromophores, sometimes fluorophores. The reason for that is if you take biopsy out of the body and have a thin section of it, a couple of microns, and put it under a microscope, bright field microscope, it looks like glass. There's no contrast to it. And that's the reason why for the last century or so, we stained tissue with chromophores after a tedious and toxic process for them to look colorful under bright field microscope so that diagnosticians can look at this is an H&E stain image of tissue. What we do here is we stop the process right here when there's a thin section label free, and we look at the endogenous fluorescence of tissue using an autofluorescence mode, standard filter sets that are commonly available in histology grade microscopes, and get these grayscale images that are diffraction limited, that contain all the beautiful morphology of the sample. And we train deep neural nets that will be enabling this transformation from grayscale autofluorescence to bright field labeled version of it by training the network with respect to images, ground truth images coming from histology labs. Once you achieve this learning, you can bypass this entire stoichemical staining and make it stain-free and virtually generating the same quality of stains, and many advantages I'll share with you in the next few slides. The technology behind this is based on a conditioned generated adversarial network. Basically, there are two different networks, deep neural nets that are fighting with each other. One of them is a generator. This is the painter. It takes diffraction limited images and virtually stains them. And this is another AI trying to understand if it's virtually stained or histochemically stained by humans. There are also some structural loss terms that regulates this. This is not a hallucinating network. This is faithful to the nanoscopic structure of tissue because at the input, we have a beautiful diffraction-limited microscopic image. At the end of this training process, nobody wins. They both lose, but then the generator is actually a beautifully trained virtual stainer. It can actually transform it, basically fooling the discriminator in the sense that the discriminator cannot sense if it's a chemical stain or virtual stain. So we have taken this framework and applied it to human tissue samples, animal tissue samples, you name it, for various different organ stain combinations. And I'll share with you some examples of what we've done. These are all blind testing results, which means these are fresh, new patient specimen, never seen by the network before, blind testing results. So for example, this is a salivary gland. The goal here is to virtually stain it with H&E. These are these are the images, grayscale input images. This is contrast enhanced for your eye. It's the same information, ANB are the same, ENF are the same. It's just showing you that there's beautiful contrast there. That's the input to the network. The network in literally seconds outputs these beautiful images of H&E, matching standard H&E that comes out of the histology lab prepared by humans. This technology works not only for H&E, it also works for more exotic spatial stains, the chemical stains. For example, this is a kidney tissue sample. The target here is Jones-Silver stain. Inputs are shown here, still DAPI, standard autofluorescence filter. Rapidly, again, within seconds, you can get the beautiful Jones-Silver stain, nicely matching what comes out of histology lab. This one is perhaps 10 times more expensive, at least at UCLA, compared to H&E. A typical good Jones-Silver stain is tedious to perform under a microscope. You need to inspect the specimen as you're staining it, and it takes approximately 3 hours to get a good Jones-Silver stain. Here, you can actually have it, a whole slide image stained within literally minutes with these patches working with a GPU in parallel. You can also do it for various other types of stains. This is a third type of stain, Mason's trichome, and two different organs, human liver and lung. Inputs are shown here, outputs are shown here. These are Mason's trichome virtual stains, and this is histochemical stains coming from the lab. The same conclusion, a beautiful match between virtually stained and histochemical stained tissue samples. This is also an expensive stain. These spatial stains are more than $100 at UCLA at least, and per whole slide. And again, this takes approximately 3 hours to stain with histotechnologists, whereas this is again whole slide within minutes. In addition to these histochemical stains, you can also do immunohistochemical staining, IHC, with this framework. Here we're showing HER2, basically an IHC HER2 as a breast cancer biomarker. This one is much more challenging to achieve because of the fact that it's molecularly specific dye, and it's even more tedious to perform IHC. The ground truth that comes from histology labs takes 24 hours to stain. Here we use 4 different autofluorescence channels with different filters, FITC, Texas Red, Sci5, and DAPI, and take these autofluorescence images and run through a trained deep neural net to beautifully match what comes out of a histology lab. Here, of course, we're much more repeatable than standard histology labs. perhaps 25% of the time, the ground truth that comes from the histology lab is actually not usable. It doesn't match the clinical history of the patient. So in that sense, the longer his technology spends with the tissue, the more problems we observe statistically coming from standard workflow, whereas virtual staining is very consistent, even for difficult to achieve stains like IHC. So these were some examples for the eye, for you to kind of visualize how you match even a complex task like IHC and spatial stains like Mason's trichome, Joe and Silver stain, PAS, and other types of stains. On top of this visual comparison, we've also given these images of virtual stained tissue samples and histochemically stained tissue samples to board certified diagnosticians, depending on their expertise, and ask them blindly, without knowing which one is which, to score various different features like nuclear details, cytoplasmic detail, extracellular fibrosis, and ask them to kind of basically score the quality of the stain. What you see here is one of those tables. We have many of these in our publications and reports, showing basically that there's no significant difference between the quality that we achieve with virtual staining versus the chemical staining. So they're on par. In fact, if you look at the numbers, we're slightly better, but it's statistically on par with state-of-the-art labs processing tissue. On top of this, we've given various different cases to board-certified diagnosticians, this time to diagnose the condition of the patient, to show again that there is no significant difference in the diagnosis performed using virtually stained tissue versus histochemically stained tissue. So you preserve diagnostic features with our virtual staining technology. But there are so many advantages. It's not just matching the standard workflow, what comes out of histology lab. There are many advantages. One of the most important advantages is standardization of the staining quality. Actually, this is something that I've mentioned very briefly at the beginning. The longer the workflow is to stain tissue, such as, for example, HER2 IHC, the more inconsistent it gets. I'm sharing here H&E results from UCLA. These are all human stained, and you can see the variation from stain to stain. And for HER2 or even other types of IHC or spatial stains, this is going to get much worse. Deep neural nets are consistent. We standardize the staining quality, which is very important for both human diagnosticians, and it's also very important for AI. So we're not a competition for downstream AI looking into pathology slides. We're actually their best friends because we will create more consistent stains for them to learn the real features of pathology as opposed to variations in stain quality. So standardization is also good for AI downstream analysis of histopathology images. On top of that, one of the most exciting aspects is we preserve tissue, and we can create from a single cross-section a panel of stains, which is very important for various different applications. For example, this is an autofluorescence image of kidney tissue section. As opposed to cutting it from different layers and getting a panel of stains, we can actually, from the same cross-section, generate nanoscopically registered panel of stains for the first time in the history of histology, basically, because every time you want to get another stain, you had to cut tissue from another section. Now you can actually have the same cross-section serving you to create an unlimited on-demand, an unlimited number of stains, if you like. And during this process, since we do not cut tissue to create these multiple stains, we preserve tissue and eliminate unnecessary biopsies. It's actually quite a bit of a problem. Oftentimes, you bring the patient back for taking some more biopsy out and for no reason except depleting tissue during staining process. So in addition to autofluorescence, which is our bread and butter for some of these results that I've shown you, you can actually use also other forms of contrast, label-free contrast, to do virtual staining, thanks to these data-driven cross-modality transformations. Earlier work, we have demonstrated that you can actually also use holography or quantitative phase imaging to do label-free imaging and transform it to beautiful H&E images of stained tissue. And this is an earlier work that literally we look into refractive index of label-free tissue, complex valued refractive index which includes the scattering of tissue and its refractive index, phase information of tissue. And that label-free information is sufficient for us to transform it to a bright field equivalent stain version of the same thing. We call this phase stain because most of the information is coming from the phase information of tissue as opposed to autofluorescence. So different kinds of label-free contrast can be used. In recent work, we have applied this to another form of contrast, and that is reflectance confocal microscopy. This is not an autofluorescence process, this is just reflectance. And different than the previous results, here we were looking into intact tissue and specific skin imaging. RCM, reflectance confocal microscopy, is basically FDA cleared for dermatopathology. The good thing about it is that without a biopsy, it can go deeper into tissue by capturing a stack of images. And we have shown that those reflectance, grayscale looking reflectance images, which is looking at the scattered light from different planes, can be actually used as the information to do virtual H&E. But this time without taking the biopsy out. And you can actually reach epidermis, dermal epidermis junction, and the superficial layers of dermis through this process and virtual stain them with a non-destructive microscopy modality that's used in clinical settings for dermatology, showing you that there are different contrast mechanisms to utilize to do virtual staining. And finally, I'll finish off by sharing with you that it's a network of, it's a highway, a network of stain-to-stain transformations you can achieve. You can start from one contrast mechanism and achieve another virtual staining task. And from there, you can actually hop on to different stains. And through this, we can also do stain-to-stain transformation, which means you can take an existing H&E stained image and transform it to spatial stains and create a portfolio of stains from existing already stained images, which is also a possibility thanks to these virtual staining networks. And you may be wondering, why should I care for this? Well, sometimes H&E is very fast, but the specialists would like to see these spatial stains. This is certainly the case for organ transplant rejection. Let's say the patient comes with a kidney transplant patient comes with some symptoms to the clinic. Well, if you're suspecting of organ transplant rejection, you start with H&E, and then if the diagnostician cannot make a call, you ask for these spatial stains to look at a portfolio of different stains. Only after then, you can perhaps make a call for the patient. This takes a couple of days in the United States, and certainly in other parts of the world, it might take a week, during which perhaps the patient is hospitalized. So you can actually, from a quick H&E, without the need for the spatial stains, create them and show the utility of stain-to-stain transformations. We have demonstrated this actually using organ transplant patients and shown that virtually stained spatial stains improve the diagnosis of the patient's condition significantly with respect to H&E only stains. So to sum up, virtual staining is a new technology enabled by AI. It saves experts labor and time, and of course saves costs, elimination of reagents. A lot of these chemicals are toxic. We literally waste millions of gallons of water every year due to these reagents, most of which would be gone if you have fully virtual staining pipeline. We preserve tissue, and we can create actually multiple stains from a single cross-section without the need for bringing the patient back for a second biopsy. And most importantly, I believe we eliminate variability in staining quality, which is very important for both human pathologists and AI. We started up a company on this in 2019 and funded by Merck Ventures and some other venture groups, together with my co-founder, Dr. Robinson. So this is actually a very exciting venture right in Santa Monica, near UCLA. And we're right in the middle of closing the Series A after the seed round last year. So with this, I'd like to conclude and acknowledge all of the students, postdocs, and researchers behind this work. Thank you very much.
Dr. Ozcan, that was very impressive. really, really exciting technology. So I'm going to open the floor. I see Ben, you have a question.
Sorry, if anyone else has one, they can go. I feel bad.
No, you're okay.
But okay, cool. Yeah, no, awesome presentation, super interesting. And I'm not sure if you're familiar with Path AI, but we invested in their seed round, so we're very much looking very closely at the space. But I guess like what I'm super interested is as a commercial endeavor, what is the target market here? I think that, one potential direction this could go is as sales to pharma, because, in pharma, especially in oncology, what they always say is that tissue is the issue. And if you can save any sort of like H&E or HER2 staining, I mean, you multiply that over multiple clinical trials, that's huge value. So I guess, are you selling to pathology clinics or are you selling to pharma? Like who's the target?
Great question. Yeah. So I think one of the lowest hanging fruits is really pharma. And that's what we have been looking into, companies like CRL, those providers of staining services in the United States and globally. We have them. I haven't shown here any animal tissue, but that's what Pictor, the company that we established, has been working heavily. Pictor in Latin means painter, so that's this is the micro painter of tissue that we have. So yes, we have demonstrated this with basically animal tissue as well, toxicology reports, drug development, saving time, and some of the stains that we have come across could take in their pipeline 2 weeks to get. So imagine, you're in the middle of generating reports for the FDA for a pharma company, right? And some of these stains, the big bottlenecks for them, not the standard H&E or other stains, these are some fluorogenic stains, they literally take 2 weeks to come. We've shown that you can do the same thing as good, if not better, in just minutes, and they see the immediate value, which means they can service more pharma companies in the same time with less resources, less bottlenecks. Especially during the pandemic, we learned something new. The supply chain for a lot of these chemicals are coming from a single country, India. And when they were shutting down there due to the pandemic, what happens to the stains? You run out of them, and the quality assurance is a problem. And that's actually one of the biggest reasons why companies like Merck and others are very much interested in this line of work, because they have a whole group looking into quality assurance of stains. And that's a painful process. And they like this technology, not only because it's fast, it can serve more customers per unit time and perhaps increase their sales, but at the same time, quality assurance will become less of a pain. And I think pandemic is teaching us that global supply chain is quite vulnerable to various different things, and you need digital technologies perhaps to help you with all that quality assurance processes that are failing.
Cool. And then one other brief question, if you don't mind, is I think that there's a lot of companies that are, waking up to the fact that there's important diagnostic information and actionable information in the slides themselves, and they're becoming a substrate for machine learning. And I think what is cool about your prediction of H&E is you do get very consistent contrast, and that is something that they're struggling with in terms of just having humans in the loop in terms of staining and the variability of contrast. I like that very much resonates. So have you ever thought about just selling your models as a service to these kinds of labs that are trying to do machine learning on top of imaging?
Absolutely. We have interest. We have some partnership with Path AI that's also unfolding, and so I think the answer is absolutely yes. And that's one of the reasons why I'm emphasizing this repeatability, because we are really not in conflict or in competition with other AI pathology companies. In fact, we are providing better images for them. And the good thing is that we can not only create more consistent H&E and other stains, but also we can give them another contrast channel that they may not have ever considered before, the autofluorescence channel. Autofluorescence is very valuable because it's molecularly specific, right? So, and you can have multiple channels if you want. So I think what is the information in autofluorescence and the virtual stain image together, and how it can make other AI companies to better stage tumors, to better detect tumors or abnormalities, etc. I think that's a question for the community to realize, and we understand our position as a digital provider of faster, larger volume and higher quality and richer information. And I think that richer information is one of the key aspects. It's not just providing the same thing, more consistent. It's actually also richer, because autofluorescence has been ignored by and large in clinical workflow. Why is it ignored? I can tell you this. There's only one reason why autofluorescence has been historically ignored, and that is autofluorescence or fluorescence microscopes were invented centuries after bright field microscopy. The reason that we have been staining with chromophores tissue for the last 150 years is nothing but because fluorescence microscopy was a late arrival to the microscopy portfolio. That's the only reason. So momentum. So, but that's going to change, and hopefully we'll be part of that change.
I see we have one more question, so I'll take this question from Dr. Akbari.
Thank you. Yeah, that's a fantastic presentation, Dr. Ozcan. Being a lab that does a lot of histology and immunohistochemistry, I just want to understand. So I understand the histology aspects and chromophore usage, but as far as the immunohistochemistry and trying to detect specific epitopes or specific proteins, right? How you went, you had a slide there where you mentioned like DAPI and a few other stains, and then you go from that to the autofluorescence, and then the using deep neural networks to detect HER2/neu or whatever other. I was just trying to understand that better, because this clearly has potential to completely transform how everybody, whether it's academic-based labs or industry or the hospital setting and pathology, but the detection of specific epitopes is what I'm confused about, and how autofluorescence, which is something that, by the way, I completely agree with you, it's overlooked. And being an optical lab, we also realize there are a lot of overlooked aspects, like scattering. Things that people overlook are actually so critical. So yeah, if you can explain that, I'd appreciate it. Thank you. Excellent.
Great. So this is a HER2, not a fluorescence-based system. We have also extended this to specific fluorescence staining, where the fluorophores, through antibody interaction, specifically label some parts of the tissue. It's the same general question of how molecularly specific antibody antigen interaction can be, stained virtually with the same framework. The underlying core, theme here is not a pixel-to-pixel transformation. So in other words, this is texture-to-texture transformation, and what we're seeing here is that with a high-resolution autofluorescence image and a sequence of autofluorescence images coming from different filter sets with different excitation and emission bands, you actually have sufficient spatial clues about where the binding is occurring. So it's actually giving you an environment information, local nanoscale environment information about the tissue and how molecularly specific events are binding. And I think it's hard for us to exactly explain that relationship, because we don't have that understanding of the tissue, its pathology, and its autofluorescence, right? But this is becoming evident to the function approximator of the neural net through data by extremely well-registered ground truth images, well-stained and well-registered ground truth images, and the autofluorescence images, that this texture-to-texture transformation is accurately representing even antibody-antigen interactions where the stain becomes molecularly specific, as opposed to something like H&E or Mason's trichome-based stain.
All right, well, excellent presentation. Thank you for the questions, everyone. So I'll move on, a round of applause for Dr. Ozcan here. Thank you. And so we'll move on to our very last presenter today, Dr. Markovich from UCLA.
Okay, hopefully you can see my slides. Okay, great. So I'm Dejan Markovic. I'm a faculty at Electrical and Computer Engineering at UCLA. I have successfully transitioned research technologies in the past. I'm a founder of Flex Logics Technologies that now employs over 85 people, and have also helped to develop proof of concept technology that became a foundation of Cerebell for epilepsy detection that is now used in over 150 hospitals worldwide. And today, I will share another fantastic opportunity in diagnostics and therapy involving deep brain structures to address enormous and growing unmet needs in medicine today. The economic burden in treatment of mental health, chronic pain, and Alzheimer's disease exceeds $1 trillion per year in the United States. There hasn't been much progress in psychiatry, particularly since the invention of opioids. Many patients spend years trying one medication after another with no relief and come to the point where clinicians have nothing left to offer. One of the most promising approaches in such cases is to use implantable medical devices for deep brain stimulation, or DBS, which so far has shown to work in Parkinson's disease and movement disorders, and has very limited success in epilepsy. Believe it or not, today's DBS devices use the same technology of cardiac pacemakers from 3 decades ago. This chest-based device that is, you can see here in the middle, for treatment of Parkinson's disease is implanted in about 10,000 patients per year worldwide. And this head-based device for epilepsy is implanted in only 300 patients per year. Both devices use 2 probes with 4 to 8 ring electrodes for continent-level access of the brain. These outdated devices focus on single targets and have no way of adjusting stimulation dosing without going to see your doctor every couple of months. This approach won't work for the vast majority of neurological conditions because the brain operates as a network where different parts interact in coordination. Now imagine that we could stimulate multiple brain locations and use recordings to intelligently access brain networks to self-adjust to optimize therapy and minimize side effects. Access to these deep brain networks is possible in surgical epilepsy monitoring, where chronic patients are implanted with many probes. In this case, you can see 10 probes with 80 electrodes all over the brain to localize seizures. These electrodes connect to an external refrigerator-sized instrument, and patients are hospitalized for 2 to 3 weeks to monitor their seizures. Hospitalization alone is $50,000 to $80,000. And when seizure can't be localized, the second stage surgery is performed to implant DBS device that you saw previously to treat seizures. Although it's an existing condition with complete insurance coverage, epilepsy is one of the most underutilized opportunities in neurosurgery. There are 150,000 new surgical cases every year in the US, but less than 1% participate. The cost would greatly reduce, and more patients would be willing to undergo surgery if this massive technology can be replaced with a small implant device that can monitor brain networks in a natural environment, at home, and over a longer period. So we developed the predicate technology under DARPA subnets program, which was the inaugural President's Brain Initiative program that provided $33 million of de-risking capital to UC San Francisco Neurosurgery, UCLA, and the Livermore National Lab for clinical and technology R&D. These are qualification units built to medical standards and FDA compliance for the DARPA program. We have an extensive IP portfolio which covers fundamental advances in sensing, stimulation, wireless power, and data. Our technology was designed specifically for interacting with the functional networks of the human brain. So we record multiple brain locations, aggregate data to a hub, and pass it down to a control module. And in response, this controller sends power and therapeutic stimulation that's individual to each patient. The key piece of implant device is our neuromodulator, with the electronics integrated with a 64 contact electrode that is cortical or subcortical. It's the world's smallest and only full duplex brain modulator. So you go away from walkie-talkies and have a two-way communication technology. And it's the size of a vitamin pill, which represents incredible level of miniaturization. Now, beyond the DARPA program, we assembled the electronics into an external clinical research system, got it through UCLA IRB, and have been testing in human participants in epilepsy monitoring with 12 human subjects so far, leveraging the clinical opportunity to do validation of the most versatile closed loop neuromodulation platform. We're able to access brain networks and also individual units in human participants. You can see here a little bit of the clinical integration of this technology that involves a lot of connectors and customization. And three things you really need to keep in mind is that this is a complex hardware and software, generally speaking. Second is that it requires extensive level of training for clinicians to smoothly operate this. And three, most importantly, it involves incredible amount of inconvenience for the subject. That's where we really need to start and work our way back. So this technology is meant to replace bulky and expensive clinical equipment that you see on the right, while providing higher fidelity neuromodulation. So this is a couple $100,000 neon codon system that we can replace while offering patient mobility with something that's on the order of 25,000. Recordings from the hippocampus from a human patient that you see here in temporal and spectral domain show concordance with this clinical neon codon system that's much larger and much more expensive than our system. We're also doing stimulations to look into stimulation response maps and build real world evidence for future FDA submission. We have this paper currently in review in Nature Neuroscience, which had 40,000 views in about a week across Twitter and LinkedIn. What we are able to do with this technology that was never done before is to enable recording of single units in freely moving humans. People were able to do this in animals before to look into play cells, but due to mobility and agility of this system, we are able to put our equipment in a backpack and have patients for the first time freely moving around. We see here clean looking individual units after spike sorting. And this is a tool that basic clinical researchers and neuroscientists are really looking forward to. We are taking this to the next step with the NIH follow-on proposal to build MIND, which stands for Miniaturized Neural Duplex, to extend the capabilities of this technology to build a dual-use externalized and implant system on the foundation of medical grade. And you can see here how we compare in terms of the stimulation, sensing, and the miniaturization abilities with respect to existing clinical systems. Each one of these companies like BlackRock, Neon Codon devices are on the order of a couple tens of millions of dollars of sales, which we think would nicely organically grow our revenues until we work out implant challenges. And you can see that we pretty much closed the whole circle, offering the most advanced full duplex and most miniaturized system out there. This is part of the bigger vision of a medical device as a service approach, where we integrate device and data for personalized diagnostics and therapy. So we start with this trial device, which we call NeuroStack, which is where we are now, and building human validation and data platform. So over time, we will build more of these data analytics and reduce the size of the implant to make it feasible for endoscopic or similar minimally invasive surgery. So this model produces 3 revenue streams. The first one is the 510K externalized device. Second one is the implant. And third one, which we anticipate to be dominant over time, is personalized data analytics. In that proposal I mentioned to NIH, we are proposing to study feasibility in 44 human participants across 2 clinical institutions, and we have overwhelming support from another dozen clinical institutions who are willing to adopt our technology. We plan to enter epilepsy monitoring as a low barrier regulatory pathway to these major new indications: chronic pain, depression, Alzheimer's. This is a highly consolidated industry, and no company presently has the solution for brain network modulation. We estimate 10 billion serviceable obtainable market, assuming adoption rates typical for DBS, which is around 7% in Parkinson's disease. So we combine modern brain-computer interfaces and data analytics for brain network modulation to address major medical and social needs. We have a great team. It's been working for years to realize this vision, and I hope you'd be interested to support this effort. Thank you.
Great, thank you, Dr. Markovich. I'll kick off the questions here. I think the therapeutic or the monitoring is really interesting. One thing that you mentioned in our prep was the cognition and memory component. Can you talk a little bit more about that?
Yeah, sure. So every time there is an opportunity in epilepsy monitoring unit, such as the one that you see here, there is a kind of dual interest. On the clinical side, we want to be able to localize seizures so we can do resective surgery for taking the tissue out. But at the same time, when patients are not seizing, then we have a whole team of scientists that come to run various behavioral experiments that are then correlated with electrophysiology to see the precise modulation of the brain networks that can enhance memory and cognition. And so that's where a lot of the specs that we developed are actually supporting this kind of unique opportunity that memory is a non-topographic and more complex than movement disorders, where you really care about individual responses of neurons that are highly selective. And so we're looking into providing features like time locking the stimulation to the natural rhythm of the theta rhythm, because science stipulates that encoding and retrieval can be kind of tracked with respect to the peak and throat of the theta rhythm. So by doing that in a kind of naturally locked mechanism, we are able to uncover a lot of science of the memory and cognition, and be able to, in the future, help with increasing our learning and various assistive technologies to help these people who have implants that we are envisioning.
And then I guess, getting into the underlying technology, I think for some of what your potential uses are, it makes sense to have it be implanted in the brain. But I think for some of the monitoring technologies we see on the epilepsy side, some of it is non-invasive. Do you see this technology being used in a non-invasive way?
So this technology, as far as the sensing goes, is universal. It can do any EXG, EMG, EEG, EKG type of technology in the frequencies of interest. This technology here is actually invasive. So this whole montage of epilepsy monitoring that you see here goes through this turban of cables that is bundled then and hooked up to an instrument. And so that's kind of the opportunity that we see, because epilepsy monitoring is a gateway. It's a golden gate to all these other big indications where scientists look into anxiety, depression, Alzheimer's. And so I think there is a great amount of opportunity that we can leverage to those kind of secondary indications to address these large opportunities in research and translation.
Great. And then my last question for you is kind of around the basic research component. So, can you talk a little bit about how your technology is used, less on the clinical side, but more on, understanding how the brain works?
Yeah, so this actually is a very important tool. So if you look into the broader scale of neuromodulation, our understanding and treatment really of these kind of, it goes from the basic science to the clinical translation to the therapy. And so far we have therapy in Parkinson's and movement disorders and cochlear implants because the tools are at the right scale. For everything else that you're talking about here, we don't even have the tools to properly diagnose indication and then be able to modulate brain networks. So in that sense, we are looking into understanding fundamentals of the brain operation and looking into generating brain response maps that is fully automated. I kind of like to use the analogy, if you have, let's say, 8 stimulation engines mapped to about 100 contacts or so, it's kind of playing a piano in the brain. You got to figure out everywhere you can stimulate what the responses look like and be able to kind of in a machine learning and adaptive manner drive the outcomes that would be of therapeutic value and accelerate the development of translational science and get more informed therapies.
Excellent. Well, I think that is all for our questions. So I want to take a moment to thank Dr. Markovich, great job. And also all of our presenters today. Everyone's working on really fascinating innovation. I'm excited to see it all move forward and, see what kind of unique partnerships and spin outs come from all of this. I'm going to hand it back to Steve to close us out.